So now we have Krishna Megh. Krishna Megh probably is known to many of you here. So he's, for those who don't know him, I think that's a minority over here. He's from the NCBS and is an evolutionary biologist and has been working with butterflies for a very long time. So today he's going to be talking to us about, generally he's going to give us insights into the field as well as then lace it with his own case studies. I believe he's going to talk about 2 case studies that he's familiar with. So with that, yeah, Krishna Megh, yeah. So yeah, maybe you want to announce the break at 3 o'clock.
Sure, I'll do that. Yeah, sure. Thanks, Emma, and thanks all the organizers actually for inviting me for this session. These are some of the things that I've been thinking about, because I just love all the evolutionary action that's going on there. And feel free to stop me anytime. I'm going to divide this in 3 sessions. The first one is going to be more of a lecture, and that's the topic that I've put up there, population to species continuum. So I'll first talk about what that evolutionary problem is with applications to taxonomy and evolutionary biology. Then we will rather take a break, maybe, that will be the coffee break. Yeah. So that will be half a, 3:30, but if you want to stop on let's move it to 3 if we can. Okay. Then we'll have a coffee break. After that, I'll start with the first case study that I want to tell you about. Then we'll take another 5 to 10 minute break, and then the second case study. This is right after lunch, so I won't want to actually assist you in sleeping better. So I'll try my best to keep you engaged. Okay, so I will start with the lecture part of this, and the background that I'll start with this is that if you look at life on Earth, you have things very simple like viruses there to something very complicated like these siphonophores, and there are millions upon millions of species that we know of already, and then many which have not even been named yet. And you have this tremendous complexity, not just in terms of species diversity, but also all the phenotypes that you see here. So biodiversity, as you know, people from India, we should be particularly interested in and intrigued about, because there is so much of it around us, and that's something that you often don't think about, but this is one of the main things that actually give us a scientific edge over many other biologists in the rest of the world. So this is something, fortunately, I was fascinated by ever since I was a kid, and this is something I've been doing as a career as well. And diversity can be viewed as different things. It can include functional and morphological diversity. Species diversity, of course, is something that you are probably most familiar with, but remember, there is a lot of diversity here irrespective of species diversity, and ecologically that's actually quite interesting. And of course, you have organizational levels all the way from genes and genomes to communities, and you can actually imagine any number of things. You could look at, for example, physiology, physiological rates. So biodiversity, depending on your interest, depending on your aptitude, you could basically take up any part of it and make it interesting. Now, when you look at all these different things and not necessarily species, here is an interesting point. Theoretically, there is no reason why you can't have a single species that shows all of the morphological and functional diversity, right? Do you agree or not? Yeah? Okay. Now, if you don't agree, let's go into exactly what are species, and we'll get to this bigger question in some time. But at least for me, theoretically, I can't see any reason why you can't have just one species, a completely panmictic population, which just shows different phenotypes and different things at different spatial and temporal scales. Okay. So this is possible, but naturally you see a lot of what we call species, and we'll get to that problem in a bit. And we, as organisms which have evolved to organize things in our minds because it's helpful to us, naturally we start putting all the biodiversity or anything really that we see around into tidy distinct biological entities in terms of biological um, biology around us, all the biological diversity around us, we try to put that into different species. Even as kids, we know ant is an ant and a spider is a spider, and so that's a very basic level of classification, but we do that naturally. Now, when you do that, you could start calling whatever we call these distinct entities as species, but then there are certain complications that arise. Now, a lion is a lion, there is no question about it, at least as far as I know. One subspecies more widespread here, but now there are questions about whether the Indian lion was really brought in here or it was naturally there and so on. But apart from those complications, there is no doubt that we are dealing with one particular kind of animal there that we will call a species. Things become complicated when you start going to more diverse groups of organisms such as parasitoid wasps and beetles and bacteria. We do not even know how many organisms are there. If you look at bacteria, only a few thousand species have been described, but if you look at any environmental sample, there are thousands upon thousands, many more thousands that are described in a single sample of soil, right, or from a single part of the ocean. So, we do know there are lots of species there. Now, there are some estimates that per species you have per species as we recognize it of larger organisms, there are at least a few parasites, at least a few parasitoids when it comes to insects. Parasitoids are these tiny insects which feed on other insects from inside in larval stages and kill them eventually. And this is a diversity which was again highly underestimated. We thought there were only a few species, a lot of these subspecies were considered to be generalist, and now more and more cases are coming up where we know that what was considered to be one generalist species is turning out to be 10 highly specialized species or more, mainly because these are such tiny things that you can't even distinguish between them under a microscope. And maybe of course the wasps know what species they belong to and what is not their own kind, but we as organisms who are very different from the wasp, we have a very hard time figuring out what is a species within that. So, whatever looked similar, and again we are looking at things which are a few millimeters long at best. Some of them are less than 1 millimeter, some of them are a very small fraction of that. So, you are looking at really tiny things. So, imagine if you looked at let us say you are an alien, you come to earth and you look at all the different kinds of humans and all the other kinds of apes, I bet it's going to be hard to draw a line, but as humans we have no doubt whether chimpanzees are our own species or not, right? And I'm pretty sure chimpanzees don't have a doubt whether gorillas are their own species or not. So, it's a matter of scale and familiarity in some sense. What you cannot distinguish, a taxonomist can distinguish. What a taxonomist cannot distinguish, the organisms themselves can. Now, imagine this problem per insect species. If there are at least a few parasitoids, and this is something that we are discovering more and more, there are specialized parasitoids that infect only eggs. There are specialized parasitoids that will lay eggs on caterpillars, which might take various amount of time, and then you have what are called hyperparasitoids, which feed on parasitoids which are inside the host, and there are hyperparasitoids in some cases. Okay. So, per host species you have at least a few parasitoids. We do not even have a good estimate of on average how many parasitoids you have, and we have about 1.678 million species. This is increasing all the time that are described so far. Most of them insects and many of them plants, and a tiny fraction vertebrates and so on. Now, if each species has at least a few parasitoids, let us say in case of insects, and each species has many parasites, how many millions of species are we talking about of just parasites, right, of just parasitoids? So, if there are about 300,000 beetles known, if there are 1.2 million lepidopterans known, and each one of those species has several parasitoids, you are looking at potentially several million parasitoids alone. Okay, and all these parasitoids have evolved in relatively recent times along with their hosts. Now imagine when you have a very large organism like a lion, with whatever divergence that you see in that species, there is still not going to be any confusion about what that organism is. When you have several millions of highly diverse groups of organisms, several million entities, you can imagine that you are going to catch a lot of them in the course of divergence, right. So, that is the setting in which I am going to ask you this question. What we call species, how we organize things, is this approach sensible? Is it reasonable? Does it really work in real life? And if it does or does not, what should we do as a matter of practicality? So, these are the three things that I am going to deal with today. First, more of a lecture, and then 2 case studies with this. Alright, so the first question, are species real? I understand this may be a little bit unfair. You are just beginning to do evolutionary biology and you are being told that there is this modern synthesis and there is this species concept and so on and so forth. In your own mind, do you think species are real? And this is not a trick question, so do not think too hard about it. What is your first reaction? Are species real? Yes? Okay, fine. Then let us go into this. So, if you think species are real, let us look at what is the most common definition of species. So, you have species which are reproductively isolated groups of populations isolated from other such populations. So, the most popular species concept is this, what is called very how should I say it? A very presumptive idea of what should be species. So, the whole idea is that reproductive isolation is a hard line. So, if you have reproductive isolation, you have distinct species. If you do not have reproductive isolation, you have something under a species category, under that taxonomic level. Okay. So, how many of you believe that reproductive isolation is a hard line? It is not a hard line? Okay. In certain cases it is. Okay. Now, we are already breaking our resolve that species are real. If this is the definition that you go with and you already say that reproductive isolation is not a hard line, then what does it mean? Okay. So, let us dive into this a little bit more. So, what we are looking at is a population, and most biologists agree that life evolved once on earth. That is a parsimonious explanation. There may be multiple origins with hybridization among those early lineages, forming what we think is one common origin. But most parsimoniously, there was one origin of life on earth, and then there has been divergence in all sorts of lineages, and then these lineages keep on diversifying into different niches, and this is what you would recognize as a species tree, what we typically see as a species tree, right. But if you really look at this, you can see this kind of structure in absolutely anything at any level. If you look at different populations of humans, depending on what alleles you choose, what genes you choose to sequence, you will get this kind of divergence. In some cases with hybridization, but still you will have fairly dichotomous branching at very small temporal scales. Okay. And this is something that you will see in Drosophila, for example. If you breed them in the lab and you expose them to different kinds of selection pressures, you are going to start getting these flies to adapt to these whatever selective regimes that you are exposing them to, and based on the genetic basis of that, you may get you may start getting divergence there. So, the rest of the genome will stay the same, highly polymorphic, but then for certain characters that matter, you are going to start getting divergence, and this could go on. You can take bacteria, and there we can actually go much further. If you have plasmids and all sorts of things which make a bacterium, depending on what is picked up from the environment and what genes are lost or gained, you may have things looking very different in a matter of years, not even millions of years in which we typically think of. So, you can see this kind of dichotomous branching or polytomous in some cases everywhere, from within population traits to across species, right. Now, the real goal of taxonomy is to find what is within species versus what is across species, and then based on whatever definition you use of a species, you will say, I'm going to call this one species, this another species, and these are two different subspecies perhaps, or whatever lineages within a species. Now, if reproductive isolation is not a hard line, and we do know now that you can get across species hybrids, you can get across genus hybrids, in some cases you can get across family hybrids, and of course genetic compatibility starts to degrade as you go further and further away in time and bring those organisms together. But for a lot of things, hybridization is real. Throughout the course, this particular course, you will hear of lots and lots of cases where hybridization is very rampant, and I will speak about one of those cases at the end of my 3 hours. So, when you start going from very recent times to older times, of course reproductive isolation is going to kick in, but within groups, especially in very diverse groups like those wasps or many plants, for example, there is no hard line. You could get hybrids, perfectly viable hybrids in populations. This has been studied in plants a lot. Some butterflies like heliconius butterflies, some mice, really well known examples, some fish, and I can actually keep on pulling examples. No matter which group you pick up, if that group has been studied to some detail, you are going to find cases where there are hybrids being formed all the time, often without any reproductive incompatibility, either between the individuals which are hybridizing with each other or their offspring or back crosses. You can look in any direction, okay. And right now, just take my word for it that you are not going to find this as a hard line. Okay, so are species real? I mentioned this biological species concept which relies heavily on reproductive isolation. There is phylogenetic concept where you say you take quite a few individuals and whatever forms what we call monophyletic lineages. You are aware of monophyly versus, okay. So, whatever forms a monophyletic group is a species, and you can of course go with dozens of species concepts that have been put out, but these two are the most common ones. Now, phylogenetists often like to push this as a better definition of species, but if you really think about it, you get these clusters only because you sample at certain levels. If you sample more, you are going to start getting into the same problem, right. And again, if you are doing a phylogeny of, let's say, big cats, you are not going to have too much trouble. You will be able to find your species as a coherent unit no matter how much you sample. There is no way lions and tigers are going to come out mixed, right. For some genes they may, for most of the genome they won't. Now, go much further down, and let's say you are working with bats or mice, and this is again, as organisms go, these are massively large and rare organisms in the ecosystem. Even mice, you may have a few million at best in an area. When you are looking at monarch butterflies, for example, you will have tens of millions in an area. So, a few million, which is again mice, usually you will probably see few 100,000, right. So, these are really large organisms on the scale of things in nature. So, even there you start to face problems. When you have so many species, you do not know again where to draw a line. You ask any taxonomist, they will agree about at least a few taxa whether they are species or subspecies or just variants in particular habitats. So when you sample those things, whether you're using biological species or phylogenetic species concept, you are going to have trouble. So as far as I'm concerned, and you may disagree with me, but we can have that conversation later. The problems of species become more hairy as you go to more diverse organisms. Okay. And then hybridization makes it more complicated. So first of all, you can't even identify, for example, parasitoid wasps, all the species that are out there. As I mentioned, what was considered one journalist species was a very famous paper several years ago in PNAS showing that this one journalist species in Costa Rica was actually several species which were specialized on one particular host, butterfly host and moth host. So this is what we are running into more and more. Now from there, imagine that there are hybridization events between these lineages. So we are getting more and more into this problem where now some lineages come together and form a third lineage or an nth lineage. So now you're trying to draw lines here, saying up to here is one species, this is another one, and this is another one, this has gone extinct, this has given rise to these two, and so on. So when you include this, it can go from genetic exchange, which goes from maladaptive to neutral to adaptive integration to hybrid speciation. So you may have just a tiny bit of gene flow between two groups. I won't necessarily call it species, and from now on for the rest of this talk we'll call them populations. Okay, doesn't matter whether in terms of taxonomy it's a population, subspecies or species. In terms of evolutionary biology, you can consider all of this as populations. Some are reproductively isolated, some are not. And of course, as a biologist, I do want to call things species. I describe new butterfly species, for example. I would not like to call them new populations, right? So there are various ways, various reasons why we would like to continue calling them calling something species. But for today's talk, it doesn't matter whether it's in terms of taxonomy, whether it's a population, subspecies or species. We are talking about divergence, and divergence across populations based on something, often ecological pressures. So this is what we are dealing with right now. So putting this in perspective, for nearly 100 years, more like 80 years, we have been focusing a lot on sympatry, allopatry and species status, and this is going back to early 1900s. A lot of thinking that we have about this goes back to that period, and this is before Meyer came up with his biological species concept, and before him actually Sewall, Wright and Fisher and others were thinking about this problem. Meyer just made this a much more cared about topic. So since early 1900s, people, mainly taxonomists, realized that there are what they called species out there, and then there were lots of small variants. So you go to one island, you find, let's say, a butterfly with one spot in a particular wing area. You hop over to the next island, there are two spots instead of one. You hop over to another island, it goes from mostly white to a little bit yellowish and so on. So taxonomists thought that these were not big enough differences to call them species, but it was still worth naming them. So they started calling them subspecies. So we went from linear binomial taxonomy or nomenclature to trinomial. So you have genus, species and subspecies, and at least for a lot of birds, mammals and butterflies, subspecies is the smallest taxonomic unit. Okay. Now we won't go into issues in taxonomy about what does subspecies mean and how to distinguish between that. We'll look at it more from an evolutionary perspective, and from an evolutionary perspective, a subspecies is a geographically structured variation within a species with little reproductive isolation within the polytypic species. So polytypy comes in when you have a single name, species name, which is carried by various entities which differ by a little bit, which we call subspecies. So, if there are no subspecies under a particular species, it is not a polytypic species or taxon. If you have multiple subspecies under it, then it becomes a polytypic taxon, easy, right. So, theoretically it is actually pretty easy to distinguish between species and subspecies. It is a geographically structured variation within a species. Now, subspecies are not necessarily on way to becoming species, they may fuse back with one another with other populations of the same species. They may become distinct, they may become extinct. There are multiple evolutionary outcomes of that. But again, there is nothing unique about that with respect to subspecies. You might get all of this in species as well, right. There are populations which are trying to diverge that you will call species, and if the ecological selection that is pulling them apart breaks down during this evolutionary time, then those populations may start hybridizing with each other. Again, whatever genetic peculiarities, phenotypic traits that they have gained to distinguish them from the other population, they just start mixing in this previously subdivided populations, and then you just get one homogeneous population, right. So, this could happen with species, this could happen with subspecies, this could happen across different populations. Now, there are some real concerns, this is especially with invasive species, at least in some cases. We know that when invasives comes, they start hybridizing with native species, and then those two start to become much more homogenized, and that is how at least in some cases we are losing native species. So, that is one very good example of how you could get this kind of moving back together of populations even across species boundaries, right. In some cases this could happen across much further genetic divergences, not just a few 100,000 years, but somewhat longer as well. So, going forward with Meyer, has everybody taken an evolution biology class at some point, or do you know about biological species concept and what Meyer said about how to define those? Yeah, okay, great. Oh, he has, great. So, since I didn't attend his classes this time, I don't know what has been taught before I came. Okay, so what do you do with allopatric populations? Do you call them subspecies when they differ just a little bit, or would you call them species? So, Meyer would call them subspecies. Most people who followed him would call them subspecies, and this is a big problem. In Indian butterflies, things which are very, very different are still being treated as subspecies, because some old British guy treated them as subspecies in 1930s when these concepts were not really clarified, and we are still continuing to follow that. So, this is a big problem. Allopatry really isn't addressed by the biological species concept, and in fact Meyer was of this very strong opinion that when you bring two species which are allopatric together, two populations which are allopatric together, if they hybridize, that does not mean anything, because you are putting them in an artificial condition, your lab, and then you are trying to hybridize them, in which case you really can't deal with this problem, right. When they are in nature, they are completely on two different islands or two habitats or whatever. So, they do not come in contact, and therefore, you have no evidence of hybridization and reproductive isolation. When you bring them together, Meyer would say no, 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 that is not right. So, then how do you decide what is a species and what is a subspecies if your definition is reproductive isolation? What about incipient species? And this is another big problem. When you are here, there is no doubt you are dealing with one species. Let us assume this is a species tree for the time being. When you are here, there is no doubt you are dealing with two species. When you are here, you are dealing with four species, and so on. What happens here? So, between this point and that point, divergence has started to occur. At this point, would you call them two species? Would you call them subspecies? Would you call them populations? Right? Now, the reason I brought up lions and parasitoid wasps earlier was that when you have, let us say, 5 large cats, the chance that you will just end up investigating this particular diverging point is very less, right? When you're looking at, let's say, millions of parasitoid wasps or bacteria or any kind of organism which is just exploding in terms of species, just by chance you're going to end up finding these situations a lot more, right? Just naturally, as a result of divergence, which is happening all the time among organisms. So, if you have millions and millions of species which are just diverging, the chance that you will end up with this is, let's say, it's a tiny fraction of the entire diversity, but just because you have millions of species, just by chance you are going to have at least a few hundred to a few thousand populations which are here, right? And now as a taxonomist you are trying to find out what is a good species, things here, and then what is here. Okay, now how many of you think that species as we define them are real? Are you feeling a little bit uncertain now that everything is absolutely a species or a subspecies? Or you're still confident? Not confident? That's very honest. Okay, so I'm sorry, I've just destroyed what was told to you for years and years and years, and do think more about it. I may be wrong. I don't think so, but keep thinking about this. That is the whole purpose of an evolutionary biology class and science. You should be thinking about things, and you should always be questioning what has been told to us is correct or not. Is that what you see with all the new studies that are coming up, all the new findings that are coming up, including things like what Mike talked about earlier, which was evolution in bursts. Hardcore Darwinists, traditional Darwinists would say that you don't have this kind of burst. Evolution happens more or less at gradual pace, and in fact if you bring up any hint of unequal rates of evolution in terms of speciation, they get pretty upset. But now you start looking at all the evidence. Another thing that most traditional Darwinians think about, and I say Darwinians because it's a caricature of what you see in evolution rather than what actually goes on, is if you say that all major morphological change is because of some genes of major effect, they don't like that. They think that evolution occurs, morphology evolves, for example, by many mutations of very small size, right? And there are lots and lots of papers written about it. In fact, I remember a paper written by Coyne and Orr in the 1990s in which they revisited this problem after nearly 150 years since Darwin and Wallace's theory, reviewing all the evidence in the 1990s about whether morphological change happens because of small changes, small mutational steps or large mutational steps. And after going through all the examples, and there were just so many examples where mutational steps seemed to be pretty large, so they were rather mutational leaps rather than mutational steps. And they reviewed all of that, and at the end they conclude that Darwin was right, evolution happens by small mutational steps. It's like, you just wrote 10 pages saying that Darwin was wrong, and your conclusion is that Darwin was right. Right? And I'm an evolutionary biologist. I believe all evolution happens. I'm not trying to say evolution doesn't happen or the entire theory is wrong. What I'm saying is that you have to be critical and always keep looking at the current evidence and what you can infer from it rather than what is being told to you. So we have gone from, within 2 slides, species being real, absolutely no doubt, to maybe not. And more complicated organisms you have, more diverse organisms you have, you are going to find more of these cases where you can't draw a line. And if you have at least several 100,000 cases like that, it's actually a pretty substantial proportion of the entire diversity that you see out there. So sure, in many cases you can draw a line saying these are species, no doubt, but as you go into complexities of divergence, keep this model in mind, then it becomes harder and harder. And this is again simple dichotomous branching in these populations, right? You will talk about adaptive radiations if you haven't already, where you get many populations which are divergent and reproductively isolated in many cases, starting from one stock. So you have all sorts of these complications, and incipient speciation is something, as I mentioned, is going to be a big problem when you go to really diverse taxa. Okay, so what people have typically looked at is, are species sympatric or are taxa sympatric? If they are, and they still show some reproductive isolation, they may still hybridize, but let's say hybrids are less fit than pure breeds, in which case they would say these are two different species. If they are completely incompatible, you would call them very well defined species, very well differentiated species. If they produce, let's say, only 60% fertile offspring, then you would say reproductive incompatibility is setting in its midway, they will become species if the course continues, and so on and so forth. So this is sort of easy. Of course, there are polymorphisms, but if you study whatever your organisms are pretty well, you will know what is a polymorphism and what is a species. Now, if they are not sympatric, it is really anyone's guess. One taxonomist like Meyer would say, if they are allopatric and differ slightly, you are going to call them subspecies. If they differ substantially, you are going to call them species. Conceptually pretty easy. What is a little bit and what is a lot? This is where fights start, right? And where two people disagree, and therefore one person is going to call them subspecies, another person is going to call them species. So whoever has the loudest voice, the most authoritative taxonomist wins, but biologically there is really no basis to that. What do we do with step clines and incipient species, as I mentioned? Does everybody know about clines? No. Okay. So if you look at any kind of environmental gradient, let's say the Himalayas, and you have a gene or phenotype frequency, as you go from, let's say, about 0 meters above sea levels to 4,000 meters above sea level, there is going to be some gradual change in whatever environmental factor that you are measuring. So if it's temperature, temperature is going to drop down. If it's amount of oxygen in there, that's going to drop down, and so on and so forth. So when you go across any kind of environmental gradient, you're going to see that things don't remain the same. So if organisms are adapting to their local environment, then you're going to see that a particular gene and a phenotype which is matched to the local environment is going to change as a function of the environmental gradient, right? That's a simple idea of a cline. So cline is change in gene or phenotype frequency as a function of some kind of a environmental gradient. So you could have a cline which is a very gradual cline in that particular environment for that phenotype, which is just straight line. Here you won't try to call individuals at this point one species and individuals at that point, because you realize that's it's just changing gradually with the environmental gradient. What happens with the green line? And then what happens with the brown line? So if you are sampling here, you are going to say, you look at a particular, let's say, a ladybird beetle. They are much darker when you go to colder climates. So when you go here, you are going to see lots and lots of heavily melanized individuals. If you go here, you are going to see lots and lots barely melanized individuals. So if you are defining your species by the coloration of these beetles, you are going to call this one species, this is another species. Now you may get, so this is called a step cline, which is what I have mentioned here. This is a gradual cline, this is appearing to be step like, and so on. This is again a continuum in the shapes. Now can you imagine ways in which you get a step cline rather than a gradual cline within the same species? No. I'm talking about when will you get a step cline in any particular phenotype, rather than, so even if the environmental gradient is changing gradually, you might get a step cline. Can you imagine scenarios in which you will get that? So if you have some thresholds, up to that threshold you get one phenotype and there is a very rapid change in the phenotypic outcome of that particular gene, let's say, or a particular environment. If you have thresholds, then you are going to get a very rapid switch to another phenotype, and that is going to produce step clines. Okay. Now you might say, sure, but then step cline will affect only one trait. Not necessary. You could have lots and lots of traits being influenced by an environmental gradient like the Himalayas, in which case if you look at 10 characters, you are probably going to get this kind of step cline in 8 characters. So if you are using this definition of, if there are lots and lots of changes across populations, you will call them species. If there are fewer changes, you will call them subspecies. Here this is not even subspecies, this is basically a step cline, but you will call this one species, this another species, right? And that's again a taxonomic problem. As I said, we are not going to worry so much about taxonomic problem, but in terms of identifying what is going on in nature, unless you actually study this, you are not going to know what you are looking at. And if you don't know what you are looking at, you might just sample in such a way that you are never going to reveal this, right? So that is the problem that we are dealing with. The nth problem that I am going to bring up is how much reproductive isolation is enough isolation, how much gene flow is too much gene flow. So if you have two populations which are completely distinct, have no hybridization, no gene flow, you will say sure they are distinctive, whether you call them species, subspecies, doesn't matter. If they have some genetic exchange versus a lot, now where do you draw a line? Okay. So I won't harp too much on this point that you really have a gradient in population differentiation. There is no real hard line in terms of species when you look at biodiversity on the whole, especially when you go further down in taxonomic gradient in terms of species and complexity. Okay, so given all of this, what are we looking at? What characters in genomic islands matter? And this is something, if you are an evolutionary biologist or a geneticist and looking for very interesting topics, here is one topic which is really hot right now. If you jump on this right now, this is going to make a name, and hopefully you are going to reveal some really cool stories. So what's going on here? For a long time people have been saying, populations diverge, and this is again irrespective of whether you call them species, subspecies. For nearly 80 years, because of Mayer's influence, we as a biologist community had been focused unreasonably too much on spatial separation. Populations are sympatric, parapatric, or allopatric, peripatric in some cases. If you don't know what they are, don't worry about it. It basically means whether you have populations in contact or not, and if they are in contact, in what sort of contact they are. So if you have a continuum from completely isolated to largely overlapping populations, then what is causing reproductive isolation? So Mayer's thinking was that because of dispersal and then isolation, you get divergence. You can either have local adaptation based on where you land up, or you could have just isolation because of time. So if you are differentiated by, let's say, 1,000,000 years, you are going to accumulate a lot of changes at random, and at some point those changes are going to become enough to cause reproductive isolation, even if you land up in the same environment, same kind of ecological pressures, right? So you could either have population divergence because of selection, or you could have population divergence just because of spatial and temporal isolation. Okay. So this spatial thinking dominated for 80 years, and since early 2000s people have been a lot of biologists, if not a large section, at least a few prominent ones have been saying, you know, we understand spatial context is important, but 80 years of discussion hasn't actually solved any problems. So let's start thinking about what causes divergence. A, what kind of ecological pressures cause divergence? Do sexual traits, for example, are they more potent than, let's say, ecological pressures? Are you more likely to get rapid divergence under certain kind of ecological pressures versus others, or when you have, let's say, things like mate choice versus male-male competition? Would these sexual traits then diverge much faster under one condition versus another, which might cause reproductive isolation? So if you do want to focus on reproductive isolation, then the question is, what are the ecological and social conditions that cause this kind of divergence? So that is turning out to be a focus in a lot of studies now, at least in some prominent labs. And now what we are trying to find out is, oftentimes you don't know what traits have contributed to divergence. What you do know is that when you look at two divergent populations, two species, what you have are a suite of characters which are different. And again, I will return to this theme when we talk about the last example, the second case study that I am going to talk about at the end. So there, the question is, what were the key characters, what were the key features which started pulling these populations apart? And that is where this genomic islands idea comes up. So if you look at any two divergent populations, you are going to find out that there are, let's say, n number of characters which differ across these two populations. Now, if you want to know what started causing this divergence, you want to find out phenotypes and genes which started to diverge first. Are you familiar with coalescent modeling or the idea of coalescence? No. So the idea is that if you look at any particular gene, any particular genetic element, and you look at all the polymorphism within that, you're going to find out that let's say this is present time and this is past. You have all sorts of variations in this, and this variation is connected by a certain structure. So each split here is a new mutation. Okay, so there are 1, 2, 3, 4 variants of that particular gene, let's say. Now, if you trace all of these, you're going to find that, you know, these two variants, variant A and 2, and this is time, remember, using some molecular clock and other estimates, you can estimate this happened at a certain time, which was more recent compared to this, and you're going to find a common ancestor of all the variation that you see here. So coalesce, do you know what coalesce mean? Coalesce is joining together, right? So coalescent here means the joining together of all the molecular variation you see in a particular, for a particular trait, in this case that one particular gene. So by coalescence we mean how far back in time all of that variation coalesces to a particular point. Now assume that these are various, you have data for something like this for various characters. Some characters, some variation may coalesce, let's say somewhere here, some variation may coalesce at that point, and so on and so forth. And if you find that certain genes, certain traits had much deeper coalescences, meaning across two populations, these started diverging much before others. Now if you had a non-diverging population, you shouldn't have that kind of a tree, right? So if you can find out what were the traits which were driving these populations apart, that is going to be what we call a speciation gene. Okay. Now whether you want to call it speciation or not, that's a different matter, but this is where a lot of genetic and evolution research is now going. Rather than trying to define species, define what is causing divergence, how is that divergence happening. And of course if you have a really cool species or group of species to show this, it's even better because you become more famous when you look at, let's say, birds of paradise or Darwin's finches or something which is inherently very appealing to a broad range of biologists and other people. So there are advantages of doing certain things, but basically this is the idea, and you could potentially do it in anything. And if you choose a more complicated group, your story is going to be complicated. It might take you some time to figure it out, but as students, I think these are the kinds of things that you should really be thinking about, which is going to make your career, which is going to first say something really big about biology, and of course you make your career. Yes. So that is true and not. So his point, if you didn't catch that, was that some people argue that there is no speciation gene. Any gene can act as a speciation gene, something that's going to pull populations apart. So if you think about it, again, don't just listen to people. Think about it, right? What's your opinion? Are there speciation genes, or is this your opinion that there are no speciation genes? So the reason I wanted that answer from you was to point out what the problem was with that thinking. So when you think of speciation genes, people think that there is a set of genes which are speciation genes. Now, what we are thinking about or talking about is when you look at divergence, like what we had on the board earlier, when things are diverging, what are the genes and which are the traits which had caused the first. So if you're interested in diversity of life on Earth, you do want to know what caused that diversity, right? So in certain kind of environment, certain kind of genes may be more critical than others. But if you know under what kind of environments and ecological pressures, what kind of genes are important, then you can start looking at those genes. And those will be your speciation gene for that group of organisms or for that kind of habitat. So it's not one set of genes that we are trying to find out. We are trying to find out when you have coalescence like this for various traits which distinguish different populations, what were the traits which actually started causing divergence. So all the morphological diversity that you see around, all the species diversity that you see around, all of that is being driven by these particular traits which are pulling things around, apart. If you don't have that, basically you're just going to have one big theoretical one species which does everything and which is everywhere. It might just look different, it might function different. So if you are going to, if you're interested in this process of divergence, what you want to find out is what is it that caused the divergence, and that is going to be your speciation gene there, right? So for example, if you find out that I keep going back to parasitoid wasps because they are so fascinating and I don't even work on them, right? So you might find that maybe there is an olfaction gene which has caused a lot of divergence. You have a mutation there, either that's going to get selected because selection already exists, or that mutation somehow just brings them into a completely new niche. For example, that mutation certainly helps these wasps, parasitoid wasps, detect odor of a damaged plant. And I'm not just pulling this out of my head right now. This is something we know right now in several species. So if that mutation actually causes this particular parasitoid to now detect odor of a different kind of plant on which a caterpillar is feeding. So this is again something that we know now from the last few decades of work. Plants produce different chemical signatures when they are damaged in various ways, and when herbivores eat them, they produce certain volatiles. And we do know there are certain parasitoids which can detect those volatiles, and then they go to those plants, find the caterpillars or whatever is eating them, and then they will lay eggs inside those caterpillars, and their larvae will then feed inside the caterpillar. So if you have that one mutation, you are going to become completely reproductively isolated certainly, right? Or you might potentially become reproductively isolated from all the other wasps that were your population earlier. So now you can imagine that this is a very critical gene for this kind of life history trait, right? So for this group, that olfactory gene or that gene family might become your speciation gene. In that context, does it make sense?
Yes.
Okay.
One
Yeah.
context it makes sense.
That is all we are talking about. We are not looking for a gene or a set of genes which are some magic speciation genes. Yeah.
So in order to be able to come up with find the gene, you would just have to look through the complete genomes of organisms.
That is where at least a lot of evolutionary geneticists are going. And it's actually not that hard anymore. You need a terrific bioinformatician, yes, but it's not that hard anymore. So if you have the right system, sequencing, genome sequencing is cheap. Making sense of the genome sequences that you get is hard. But if you have the means to do it, actually that is where a lot of labs are going. There's no other way to do it, right? Yeah.
About speciation, ultimately to understand the diversity and all the evolution that happens that led to the diversity phenomenon. So that idea is that you will have to come up with a pattern to explain all the diversity, and that is one purpose of describing speciation. So for so many millions of groups of organisms, every setting can be different. So will we ever find some common pattern? And if it is
Yeah.
one gene or some group of genes for every different organism, so then everything is a speciation, right?
I had put this point, I have put it in one of the later slides, and we'll come to that later. So can you just bring it up then? So his point is that we are looking for patterns, and if under various ecological conditions for different organisms, if you have different genes, then everything becomes a speciation gene. But not everything becomes a speciation gene in every organism. As I mentioned in wasps, that olfaction gene might become a speciation gene. In a set of plants, a flower color gene might become a speciation gene, or something that produces a distinctive odor of flowers might become a speciation gene there. Now from there, whether you can draw generalities, that is the point that I had put at the end, and we'll come back to that. Okay, so going from a lot of confusion, we are again coming back to some interesting things that you can do with this kind of diversity that we started with. Now, there are some, because of all this discussion, people have started looking at various interesting things that we can do and interesting ways in which we can distinguish various phenomena that are happening out there. One of the cool ideas is distinguishing between hybrid zones, between species and subspecies, if you are really interested in looking at that kind of a thing. And a lot of work has recently happened since 1980s on heliconius butterflies, where James Mallet and some of his colleagues have looked at frequency of hybrids, and this is what you see. So this is again bringing back somewhat slight order in groups of organisms which are complicated, but not so terribly complicated that you can't really draw lines. So this is what Jim Mallet and others have done. This is, since this work was on heliconius butterflies, I'll again use an example of butterflies. Let's say, based on the colors here, there is a blue butterfly and a green butterfly. This is species character. You can have any character. We are going to call this butterfly wing color. You have some distribution around the mean, and this is frequency of that particular phenotype. So you have one blue species, one green species. There may be some overlap, but you could also imagine two populations which are completely apart. There is absolutely no overlap between them. Okay. So if you have, when they come in contact, when the blue and the green butterfly come in contact, if you have what is called a hybrid swarm, you would call that a subspecies, a reproductively non-isolated population. So what is a hybrid swarm? So when these butterflies and these butterflies mix, they don't discriminate between green and blue. They will just mate with anything that's out there. Okay. So with F1 you are going to get some proportion of things that are in between, but you are still having largely A and B. With F2 and Fn you are going to get more and more and more mixing, as a result of which you are not going to have too much at the ends, you are going to have something in between because of constant hybridization and recombination, right. So hybrid swarm is a population which does not show any characteristic of either of the parental populations, okay. And this is a characteristic of two populations which are not reproductively isolated. Whereas if you have a situation where so all the dashed lines are what you see in hybrids or in hybrid zone. So when these two things come together, you may have some mixing, because of which you have the two distribution come slightly closer, but still most of the population is either blue or it's green. So instead of this much overlap, things that were intermediate between these two, now you have this much overlap in the two distributions, but you still have largely two distribution. You have a bimodal distribution of phenotypes, right. So even when these things come in contact, if they maintain more or less their genetic genomic trait combination integrity, then you will call them species. And if you have a hybrid swarm like this with one peak in between, you will call them subspecies. And if you buy this picture that I am trying to sell or tried to sell in the first two slides, then it is whether you call them populations or not, I mean species, what is reproductively isolated or not, you should get this pattern, right. So this is without reproductive isolation, this is with reproductive isolation, but some hybridization. So in this case, reproductive isolation is not an absolute hard line. You might still get hybridization. Hybrids may even be fertile, for example, but they may not be particularly viable, right. They may not be particularly adapted to the environment, or there may be just very little hybridization between the two original populations themselves. So whether it's pre-reproductive or post-reproductive isolation doesn't matter, but the fact is that you get this versus this pattern. And Jim Mallet has particularly been pushing hard on this kind of distinction between what are species and what are subspecies. Another thing that a lot of people are interested in, and Mike actually is much more in this school of thought, which is understand whether hybridization is happening and what are its consequences, rather than trying to define species and subspecies as other people have been trying to do. So it doesn't matter at what level you are talking about hybridization. What matters, what is interesting is if there is hybridization, what is causing that hybrid event and what are the evolutionary consequences of that. So David Hillis, who is one of the most famous living phylogeneticists, he likes to give this analogy. Let's say you have two rivers, they start from two different parts of the mountain, or in case of South India you can say east flowing and west flowing rivers from Western Ghats. Now if you take a bucket of water from these rivers and pour them on the other side, do they become the same river? No, right? It's just a bucket. Now if you link them, we have massive ambitions of linking rivers. If you link them, do they become the same river? No, the water mixes, everything mixes. Anyway, again, this is not a trick question, don't think too much about it. The point is that you may have something from buckets of water going between rivers to two rivers completely merging. in which case they may have some, and again rivers keep flowing, whereas we are talking about genetic exchange. So in that sense you may have two independent origins of whatever genetic diversity you are talking about, but with hybridization, if they all come together and become one reproductively integrated population, then it becomes one evolutionary entity rather than two. So when you look at this entire spectrum, why is hybridization happening and what are its evolutionary consequences, both for adaptation to a particular environment as well as evolutionary consequences in terms of whether these entities remain still distinct but exchange genes that may be particularly critical for adaptation or not, right. So this is another very interesting evolutionary field where you might actually make a lot of contributions, irrespective of how you look at biodiversity. Yes, you have a question?
I just wanted to ask, so when you have these hybrids between, say, these two butterfly species, which are quite distinct, so if we say that some of the hybrids are surviving and they are fertile and viable, and then you have their offspring, and supposing the two parent plants were having specific host plants, then what is it that the larvae feed on? Do they feed on species A host plants or B host plants?
So that's another very interesting area in which a lot of work is happening. So if you have host plant adapted populations, when they hybridize, what happens to the kids, right? Do they feed on one species of plant or the other, or they feed on something completely different? And there is no single answer. You might find different answers in different systems. In some species hybrids, you might find that the hybrid population feeds on something completely different. You might find situations where the hybrids feed on nothing and they just die, and you might find situations where the hybrids don't mind this or that host plant. So there is no general pattern there. It really probably depends on chemistry of the larval host plants and olfaction and probably lots of other things. But yeah, that's another interesting set of questions that you can investigate, and that becomes really important with agricultural productivity and vulnerability and things like that. Okay, so having talked about evolutionary biology a little bit, I will talk a little bit about taxonomy, but very little bit. I'm really not going to go into too much detail. I'm going to go into detail only as much as is needed for the evolutionary questions, one case study that I'm going to talk about. So, our subspecies products of poor taxonomy. So, there has been too much emphasis on geographical and reproductive isolation to define species and subspecies, as I mentioned earlier, which has sometimes misled efforts to name and rank taxa. There really shouldn't be a fight. If you are an evolutionary biologist, or at least the kind of evolutionary biologist that I am, I really don't think there is any need to accept that sometimes you are going to have gray zones. Some people might call something a taxon at whatever level, others might just say, oh, this is variation. Some might say this much reproductive isolation is okay, that much reproductive isolation is not okay in terms of defining species. But there is no doubt that you do have this evolutionary process. So, understanding that evolutionary process and all the cool things that are going there in pulling things apart, or sometimes bringing traits across groups because of hybridization, that is what is of interest scientifically. But apart from that, what you do have when you do want to put a name there is biology of relevant taxa is often poorly understood. So, actually what time is it? Is it time for coffee break?
Yes.
Okay. So, I will quickly finish this slide, I think, and after that I have case studies coming up. So, biology is often poorly studied, understood for a lot of things to actually say that we know something is reproductively isolated or not. Also, this is lagging way behind naming variations. So, in particularly well studied groups like birds and butterflies, almost everything that's out there has been collected at some point, and almost everything that's variable has been named at some point. which is why you have 15 synonyms for one species for a lot of birds and butterflies. So because biology, understanding of biology lags a lot behind naming. You can you can name a population in a matter of days, whereas people spend 40-50 years studying something complicated like Darwin's finches, which is what the grants have done. So there you have a problem, and sometimes subspecies or species may be real, sometimes they really may not be, and this is the point that I am going to stop with. Everyday examples of this abound. So if you, even if we say that in some groups of organisms species are real, because you don't find too many populations which are trying to diverge at this node, and therefore defining species is going to be easy, or at least defining clusters is easy. Where you draw a line still is hard, which is why I've specifically said sometimes subspecies and species are real, sometimes they are not. So what do I mean by that? This is one famous case that we published last year, not famous actually, this is one interesting case that we published last year. So you have two species of what we call Nawab butterflies, they are under Caraxes now. In India there were two species, one called Athamus, one called Agrarius. Agrarius was considered to be a polytypic species. There were several subspecies in Southeast Asia, and for Athamus it was one subspecies which was distributed from India all the way to Southeast Asia and southern China. So one really largely broadly distributed subspecies with some more subspecies in islands in Southeast Asia, and then you had one species which was considered to have several subspecies across biogeographic breaks. So what we found out was that what was considered to be one subspecies, one species Agrarius, was actually 1, 2, 3 and 4 clusters, and then what was considered to be Athamus was 2 clusters, and then you have other species. So what was considered to be this polytypic species Agrarius turns out to be 4 distinct populations. Now if you do not want to call them different species, then you will have to call this entire thing as one species. Okay, if you know how to read phylogenies, that is what you will have to do. Either call these 4 allopatric populations distinct species which were earlier considered to be subspecies, or call this entire species group just one species, because of the way monophily works here. And we ended up calling them all different species to keep monophily of things that here where people say that there is no doubt that these are distinct species. So if you don't want to merge those distinct groups, then you have 4 species, and we have one one species in India, Agraria, which was the nominotypical subspecies as we call it earlier. And this is another interesting example. This is recently worked out by my postdoc Zanvi Joshi. Some of you might know her, and this is one group that we have looked at, Argenis hyperbius, and again you have 2 what we called subspecies in South India which are turning out to be distinct from hyperbius. And I won't go into too much detail of that, but basically when you look around, it's not necessary that interesting things happen somewhere else. You can do interesting things in your backyard. We have so many species with so many interesting biological problems. You can pick up anything and you're probably going to find something interesting there. So this is a common species. If you go to Ooty, for example, for sure you will see this species, and we are finding that there is biologically something interesting going on there. And if you look at divergence here, this is taxon level divergence. You pick up any 2 taxa in this phylogeny and look at what % divergence you have, and this is what you see. For different genes, you have that much divergence between what is hyperbius and what are other things. So of course mitochondrial genes are more divergent than nuclear genes, which is what there is something you do expect. And then whether you call them species, and if you don't, then at what level becomes a problem. So this is 2%, and there is this golden rule that taxonomists like to follow. If something is less than 2% divergent, then it's the same species. If something is more than 2% divergent, then it's these are different species. There is no logic behind this, but some taxonomists like to use this. So by any logic, you can't really draw a line here, and this is one interesting case. Alright, so I'll stop here, we'll take a coffee break. I must say that I have seen very few people dozing. What's up with you guys? After lunch you should be dozing off. So maybe you find this interesting, but we'll still take a coffee break, come even more refreshed, and then we'll continue with the two case studies that I want to talk about. Alright.